All right, so chapter 10, section 5, we're talking protein functions. Now, you can have lots and lots of different proteins inside of the cell because you have so many different amino acids, and we can put them into different orders and we can make them different lengths. We can have lots of different primary sequences, which will fold into different secondary sequences, and so on and so on and so on. And so you end up having proteins with huge number of functions inside of the body, right? You can have messenger proteins like insulin. If insulin is secreted from the pancreas, that tells us that our blood sugar is high, we've just eaten, and it'll tell the other cells, hey, you can take the glucose in from the bloodstream and bring it into your cell, right? So that's a messenger. But you have to have something that insulin to bind to. So we have receptor proteins on the surface of cells that bind to the insulin and say, oh, insulin's here. Okay, I'm going to do this cascade of events, which is then going to allow glucose to come in. Well, if you want glucose to come in, you have to have something that's going to transport it in. So you have a glucose transporter that's going to transport glucose from outside the cell in the bloodstream to inside the cell. Then you also have things that have to transport things like oxygen, right? Things we need to survive. An energy source like glucose, and we need oxygen. So what's going to transport oxygen through our bloodstream? That's hemoglobin. So maybe you've taken things in and now you need to store them. So in order to store nutrients, you have a couple of different things. You have ferritin. Ferritin will store iron inside of our cells because iron is a really important ion inside of the cells. Casein is a storage protein in milk. Gluten, we talked about, as a storage protein in grains. My favorite, contraction requires two proteins, myosin and actin, and myosin and actin make up the contractile units inside of muscle cells that allow our cells to, contract, and then you get, muscles if you actually had muscles. Protection. Antibodies. Oh my gosh, antibodies are so important. Antibodies are always on the hunt. You should have certain things inside of your body that make you, that make me Dr. B. And if anything comes into my body that is foreign, then antibodies should bind to those foreign substances. And that's like a red flag that says, hey, immune system, you need to come and break these down, eliminate them, get rid of them, right? You have things that maintain the shape of our cells, like collagen, right? We said you have to have certain collagen inside of your skin so that you don't get wrinkles. Actin is also a supportive structure. It's like the inside skeleton of a cell, and it helps the cell maintain a certain shape. So we call it the cytoskeleton, cell skeleton. And then you have enzymes that function in catalysis. So these help our biochemical reactions go at a pace that is quick enough that we can actually do anything. So for example, pepsin is an enzyme that will actually cut up peptide bonds in proteins that we eat that are in our stomach. And so that helps us with digestion. So there are lots of different functions. When we talk about communication between cells, we really need to talk about three different things: messengers, receptors, and transporters. So how do we get the message from one cell to another cell? Well, we need something like a hormone. A hormone is a chemical. Sometimes it's a peptide, sometimes it's a big protein, sometimes it's other things, but it's created in one part of the body and then it affects another part of the body. So if you make insulin in your pancreas, well, insulin can actually work on just about every cell in your body. So it'll act on your brain cells and it'll tell your brain cells, hey, there's glucose, and they can take up glucose. Receptors always sit basically inside of the membrane, and they face the outer surface so that they're sitting inside the membrane waiting, right? They're waiting for a signal, and that signal is something like a hormone. And when the hormone fits inside of the receptor, then the receptor will make a change in the bottom part that's sticking on the inside of the cell, and it will tell the inside of the cell, hey, start doing whatever cascade of events inside the cell that need to happen. And then a transporter is another membrane protein, and that membrane protein must span the entire length of the plasma membrane. So it has to be able to take a molecule that's outside of the cell and transport it all the way across the membrane and get it into the cell, like glucose. Hemoglobin was one that we talked about, and that's going to transport oxygen. And so this is one that requires a quaternary structure because you need 4 subunits of hemoglobin in order to have biologically active function. So how are they attached? How are those 4 subunits attached? Through hydrogen bonds, through London dispersion forces, and through salt bridges, right? And so each of the subunits, each of the 4 peptides contains what's called a prosthetic group. So it contains heme, which has iron, right? You know, iron will rust, right? Well, what is rust? It's iron binding to oxygen in the air. And so, well, what happens to iron if it's inside of hemoglobin? Well, it still wants to bind to oxygen. So the iron's going to bind to oxygen. And so there are 4 heme groups. So you kind of have 1, 2, 3, 4 heme groups that are able to bind oxygen. And so each one will bind 1 O2 molecule. And so each hemoglobin itself, each protein, each quaternary structure of the protein can transport 4 O2 molecules at once. And so you have a different conformation of the protein depending on if it's carrying oxygen or not. And that allows oxygen to disassociate when you get to certain parts in the body. You want to drop it off, right? And there are certain parts of the body you want to pick it up. So I want to pick up oxygen in the lungs, and then I want to take that oxygenated blood and I want it to go all the way down to, let's say, my toe, and I want to drop it off. And so when you pick up the O2 in the lungs, you'll have one conformational change. And when you drop off O2 in another part of the body, you'll have a different conformational change. And so that's the oxygenated and the deoxygenated state. So oxygenated has O2 bound, deoxygenated does not. But a lot of times you'll see that in lots of different proteins, that they change conformation based on presence of something. So antibodies are so cool. So your body is supposed to be on the lookout for things that are foreign, and we call those foreign things antigens. And those antigens can be bacteria, they can be viruses like COVID. And your immune system will produce antibodies that will recognize specific antigens on the virus or the bacteria. And so the antibody actually consists of 4 polypeptide subunits. There are 2 heavy chains and 2 light chains. So if I can do this, let's see if I can highlight. So this is 1 heavy chain right here. Here's a second heavy chain. Then you have a light chain, 1, 2. So those are our 4 subunits. They're not exactly the same. They're actually a little bit different. 2 of them are long and 2 of them are short. But when you look at this, the secondary structure, right, has all kinds of beta pleated sheets that are stacked one on top of the other. So it's kind of hard to see, but like there's a beta sheet, there's a beta sheet, here's a beta sheet, here's a beta sheet, here's a beta sheet, right? They're all stacked one on top of the other. And then, so that's our secondary structure, right? But we're going to have to hold together the secondary structures to make a quaternary structure. And that's like one color. So that's all my green is a quaternary structure. But then I need to link my heavy chains and my light chains together. So I'm going to make my tertiary structure, and then I get a quaternary structure. So our quaternary structure is held together by disulfide bonds between the polypeptide chains. So what you see here, you see these SS, SS, SS. Those are all disulfide bonds. Disulfide bonds, right? And so when you look at an antibody, what you're going to see is it's kind of shaped like a Y, right? So the very ends of the Y here and here are called the antigen binding sites. So these are the sites that vary depending upon what virus or bacteria it's targeting. So it gets very, very specific. But the other portions of the molecule, like areas down here, are very general. And so your immune system can recognize them and see lots of aggregates and go, whoa, something's not right, and mount an immune response to it. Okay. I think that's it. Yeah. Okay. So let's go do a couple of questions. Not too bad today. So we got a few questions that we're going to answer about protein functions. So we need to match each protein in column A with its function in column B. Should be pretty easy. You should pause it and try it on your own. So collagen, remember collagen we said was in our skin, and that really helps to, ooh, I'm going the wrong one. All right. So it really helps to maintain the structure and maintain our skin without wrinkles and all that kind of stuff. So this is a structural connector. It's something that that provides support, right? Hemoglobin we talked about as being an oxygen transporter, something that carries oxygen. So we have hemoglobin and we have myoglobin. Hemoglobin's in our blood, myoglobin's in our muscles, right? Antibodies are there to recognize and bind to foreign substances in our body. And the last one we said casein was a storage protein, right, in milk. All right, let's see. 10.39, the glucose transporter provides facilitated transport across the cell membrane. So glucose is going to go right from, if this is the plasma membrane, so we have outside and we have inside. And so remember, it's a bilayer, right? And we have all those hydrophobic tails pointing inwards, and I'm not going to worry about drawing every single one. But what you want is for this glucose transporter, right, in order to, let's see if I can do this, to span the entire membrane and allow our glucose to get through, right? And so we have glucose molecules that are out here that need to go in here. So do you think this transport is active? A, after you eat a meal. What do you think? I would say yes, it is active. And support your answer. So why is it going to be active? Well, it's active because we have high blood sugar. If we have high blood sugar levels, well, our body's going to make insulin. So body will make insulin. And when our insulin binds to those receptors, right, it's going to activate our glucose transporters. Right? And then they can bring them into this, they can bring the glucose into the cell, and we can get ATP basically made from that glucose. All right, B, after you wake up in the morning, I would say this is inactive, right? Because we have low blood sugar. We haven't eaten in a long time, right? We have not eaten. So we don't have the glucose to spare. So we actually are functioning on reserves of glycogen, right? And our body's saying, hey, be very careful. Oh, and my screen sharing is paused. Oh, okay. So that's okay, right? So just go back and check. I'm sorry. I don't know why my screen sharing paused. But this is what, this is where we were, sorry. But so after we wake up in the morning, we have very low blood sugar. We cannot take in glucose. If we took in glucose, if all of our cells took in glucose after we woke up in the morning, we would pass out because our blood sugar would get way too low, right? All right, let's see. 10.41. If you come in contact with a virus like influenza virus, proteins on the surface of the virus act as antigens, and the immune system produces antibodies to mark the infecting virus for removal. Works for all viruses. So which part of the antibody binds to these surface proteins? Well, remember we said that our antibody kind of looks like a Y. There's our heavy chains, and then we have our light chains, right? And so it's at the end of the combination between the light chains and the heavy chains, and this is called the antigen binding site. Right? This whole thing is an antibody. And antibodies recognize antigens. Well, what part of the antibody recognizes the antigens? It's these antigen binding sites. All right. Have a good rest of your day. I'm sorry I messed up the screen share.